biology 101. That's my expertise, anatomy professor. Teaching with ease, I'm breaking it down from head to toe. I'm about to drop some knowledge, yo, here we go.
Alright, so we left off talking about lymph nodes, which are these small lymphoid organs from anywhere from a millimeter to about an inch big. They look like a kidney bean. You don't need to memorize the path through a node. What I want you to know is that lymph is going to come in as traveling down these vessels, and they're going to enter the node and become what we call afferent lymphatics, carrying lymph to the node. And the lymph will circulate through the node. You'll notice all these germinal centers where they have B lymphocytes. There's other types of immune cells in here too, and eventually lymph's going to leave the node at the efferent lymphatic, which is found in the hilum. Hilum is just an indentation. It's where blood vessels enter and leave the node along with nerves. Another organ we need to talk about is the thymus. The thymus is going to produce hormones that are important for developing functional T cells. The hormones that the thymus makes are called thymosins. After puberty, the thymus is going to shrink and it becomes fibrous and fatty. This is a process known as involution. And this is part of why you're more susceptible to diseases as you age. This is the spleen, which is right here. In this diagram, you can imagine that the patient's laying down with their feet facing towards you. So here's the spine right here. So this is the liver. This is the left side of their body. This is the right side. So the spleen sits on the right side, just behind the stomach. So it's here. Now the spleen has the largest collection of lymphoid tissue in the body. It essentially performs the same task for blood that the lymph nodes perform for lymph. And it does three main actions. Number one, it removes abnormal blood cells by phagocytosis. It also stores iron that gets recycled from red blood cells. And the last thing it does is it initiates immune responses to antigens that might be circulating in the blood. Now, the spleen is surrounded by a fibrous capsule, which you'll see on the outside here. The cellular components within the spleen make up what we call the pulp of the spleen. So all this inside, that's pulp. There's red pulp and there's white pulp. The red pulp is going to have red blood cells. So anything colored red here has red blood cells. But the white pulp, which you see here, it's more lightly colored. It's closer to lymphoid nodules and it's full of lymphocytes. And that's all that we need to know for the lymphatic system, along with what we discussed last time. And that brings us to our next PowerPoint, which is the immunity PowerPoint. All right, got the immunity PowerPoint up. So the thing about the immune system, humans have two types of immunity. Number one, there's innate immunity, and this is non-specific. It's general immunity, so it's looking to get rid of anything it encounters. The adaptive immunity is the other one, and it is highly specific. So it's looking to destroy things that it recognizes, and only those things. This will make more sense in a little bit. So, the innate immune system prevents or slows the entry of pathogens, or it attacks them if they do enter. In order to cause issues, a pathogen has to enter body tissues, i.e., it has to cross an epithelium. This could be your skin, this could be your mucous membranes. And so the integumentary system provides the major physical barrier to the external environment. Physical barriers prevent pathogens from entering tissues. So you have the epithelium itself, which is multiple layers of protection. Additionally, you have these desmosomes, which, if you remember, these tightly link cells together. You also have hair, and hair protects us from abrasions. It can prevent hazardous materials or bugs from touching our skin. You also have secretions from sebaceous glands, which are going to flush the surface of your skin, wash away microorganisms. Some secretions from sebaceous or sweat glands even have antiseptic chemicals. They can even have antibodies. And the mucous membranes lining our tract systems also protects us. And malt helps there as well. Also part of innate immunity are phagocytes, which respond to pathogen invasion. Phagocytes essentially serve as both janitor and police officer in the peripheral tissues. They remove debris, they respond to pathogens that have breached the barriers, and oftentimes pathogens are simply removed by phagocytes before the lymphocytes even detect them. They all function in similar ways, the different phagocytes, but what they target can change. So if you remember, phagocytes include neutrophils, eosinophils, and cells of the monocyte lineage, i.e. your macrophages. So neutrophils, as you know, target bacteria or cellular debris. Eosinophils are going to target foreign substances or pathogens that have been coated with antibodies. A lot of these are allergens. And there are two major classes of macrophage. You have fixed macrophages, which are permanent residents of specific tissues. They go there and they stay there, and that's kind of their patrol area. Free macrophages, on the other hand, travel throughout the body. Another part of the innate immune system is the natural killer cells, and NK cells perform immune surveillance. They detect and destroy abnormal cells, and that includes abnormal self cells, and it also includes non-self cells when they appear in the peripheral tissues. So for example, the plasma membranes of cancer cells contain proteins called tumor-specific antigens, TSAs, which natural killer cells will detect, and then it'll destroy those cells. They do this by first recognizing the cell as abnormal, and then they adhere to it. Then the Golgi apparatus, which, if you remember, packages proteins and lipids, will rotate around the nucleus. Rotates around the nucleus until it points at the abnormal cell. It's kind of like the Death Star in that way. The Golgi is then going to produce a whole ton of secretory vesicles filled with a protein called perforins. The perforins are going to be released by exocytosis, and they're going to poke holes in the abnormal cell. These holes are called pores, and the pores allow ions and proteins, etc., to exit the abnormal cell, and at that point, the abnormal cell loses its internal integrity and just disintegrates. So mistakes are often made during cell division, and natural killer cells help to ensure that while abnormal cells, they usually undergo apoptosis when there's a mistake in cell division. But the ones that don't undergo apoptosis, natural killer cells make sure that they don't survive and cause cancer. So cancer cells often mutate to avoid detection by natural killer cells. This is called immunological escape. And sometimes tumors have a surrounding capsule even that will protect them from being detected. Sometimes the cancer cells just don't make tumor-specific antigen, and so they can't be detected. Sometimes the cancer cells can actually fight back and kill the natural killer cells. And either way, when escape occurs, it allows the cancer to metastasize and form secondary tumors, which is a bad thing. This is why cancer isn't really one disease. It's actually, dozens of diseases, and part of that is how the cancer cells mutate and participate in immunological escape. So this is a big scary slide, and the good news is I don't want you to memorize almost any of it. Here's what I want you to know. There are these things called interferons. They're shown here on the left side. You have 3 kinds of interferons. Interferons are small proteins. They're proteins that we call cytokines. A cytokine is a chemical signal that coordinates cell activity. And interferons are released by activated lymphocytes, macrophages, or cells infected by viruses. And these interferons bind to surface receptors on normal cells, and they trigger the production of antiviral proteins. These proteins don't prevent viruses from entering cells, but they do interfere with viral replication inside the cell. So interferons slow the spread of viral infections. They also stimulate macrophages and NK cells. There are 3 that get discussed a lot. There's alpha, beta, and gamma. Alpha is produced by viral infected cells and stimulates NKs. Beta is secreted by fibroblasts and slows down inflammation. And gamma is secreted by T cells and NK cells, and gamma stimulates macrophages. So just remember, viruses, when they infect cells, viruses aren't able to reproduce on their own, so they get inside our cells and use the machinery inside of our cells in order to make more copies of the viral genome. The rest of this, you don't really need to know. This is related to the complement system, and I'll just tell you a little bit about the complement system. Your blood plasma has over 30 proteins, and that's the complement system. They complement the actions of antibodies. These proteins will interact with each other. They form these complicated cascades that are similar to blood clotting. There's 3 pathways involved, but the take-home picture is that the complement system helps to kill pathogens and stimulates inflammation as well. Speaking of inflammation, so inflammation is a local tissue response to injury. It produces redness, swelling, heat, and sometimes pain. Lots of different things can produce inflammation regardless of the cause. Somehow the cells are killed or injured, connective tissue's damaged, or the tissue's injured in some other way. The idea behind it is that it limits the spread of injury or infection, so it just limits the injury. And here's some things associated with that. You don't have to memorize that. Just know what I just said for the exam. Another thing that's kind of inflammation adjacent is fever. So fever is a body temperature greater than 99 degrees Fahrenheit. Some people might say 100 degrees Fahrenheit now. And you have these proteins called pyrogens. Pyrogens can reset your thermostat, which is located in the hypothalamus of the brain. And when pyrogens send their signals to the hypothalamus, your body temperature goes up. Fever can inhibit pathogen growth and spread, but most importantly, it increases your metabolism by about 10% for every degree that your fever goes up. And this mobilizes more resources in your cells to deal with the problem. So these are all of the factors that are involved in the innate immune system. You have physical barriers designed to keep pathogens out. You have phagocytes, which just generally kill things that shouldn't be in your body. You have immune surveillance carried out by natural killer cells. You have interferons, which are involved in fighting viral illnesses. You have the complement system, which is a bunch of proteins that complement antibodies. And in fact, the proteins of the complement system themselves can lyse pathogens. You have inflammation, which will limit the spread of infection or injury. And finally, you have fever. And when you have a fever, you increase your metabolism, which allows your body to mobilize more resources to deal with an infection. And a higher temperature also can sometimes inhibit viral or bacterial growth. So now we're talking about adaptive immunity. So we know that innate immunity is nonspecific. You are just generally getting rid of things that don't belong or keeping them out. Adaptive immunity is specific, and it's produced and coordinated by B cells and T cells. So you'll see here there's adaptive specific immunity, innate non-specific immunity, and remember, immunity is the ability to resist infection, illness, and disease. Now, specific immunity can be acquired through different ways. Most commonly, you are familiar with immunization, which induces an immune response. Adaptive immunity can be active or passive, and you acquire specific immunity to an antigen only when you are exposed to it or you receive antibodies from another source. So active immunity involves exposure to an antigen. It can be natural. If it's natural, you're exposed out in the environment, so you catch an illness, you get a cold, and you'll develop immunity to that specific cold virus. It can also be artificial, where you are administered the antigen to prevent the disease, and this would be a vaccine, artificial active immunity. Passive immunity is produced by transferring antibodies from another source. It can be natural. In this case, the transfer of maternal antibodies across the placenta or in breast milk, or it can be artificial, which would be the administration of antibodies directly to your blood to combat infection. So innate immunity is simply genetically determined, but adaptive specific immunity requires exposure to an antigen, generally speaking, unless you have passive adaptive immunity, in which case you are given the antibodies in some way. Regardless of how you get it, adaptive immunity has 4 properties: specificity, meaning for every antigen there is an antibody. T cells and B cells bind to one target, and they will ignore all the others. There's versatility. So there's a large diversity of lymphocytes sensitive to different antigens. When they're activated, they will produce clones. There's memory. So if one daughter cell attacks an invading pathogen, the other is inactive until they encounter the antigen again. These inactive memory cells allow for pathogen response at a later date, so the immune system remembers an infection you've had, and so in the future they're better prepared to deal with it. And finally, there's tolerance. So the immune system ignores your body's own cells, but it will target foreign cells. So immune system tolerates the cells of your body that are supposed to be there. Specific defenses are not activated until antigens are presented on infected cells or on phagocytes, and this is called antigen presentation. Now, antigen presentation occurs when an antigen glycoprotein combination, some complex with an antigen on it that is capable of activating T cells, appears in a plasma membrane. Now, the structure of the glycoproteins that the antigens are attached to is genetically determined. And the genes are found on chromosome 6 in a region called the major histocompatibility complex, or MHC. So the proteins are called MHC proteins. And class 1 MHC proteins are in the plasma membrane of all nucleated cells, and it's part of how our immune cells can find and eliminate the bad cells. So looking here at MHC, remember class 1 MHC proteins are in the plasma membrane of all nucleated cells, and class 2 MHC proteins are only found in the membranes of antigen presenting cells and lymphocytes. So that includes all of your phagocytic cells like macrophages, as well as dendritic cells. Class 2 MHC proteins only appear when the cell's processing antigens, but class 1 MHCs basically on all nucleated cells. And here's the way it works. So you have a virus that infects a cell in your body. This will trigger antigen presentation by class 1 MHC proteins. So the infection results of these abnormal peptides in the cytoplasm, and then the endoplasmic reticulum is going to produce class 1 MHC proteins to which the abnormal peptide, this antigen, gets attached. And this will go through the Golgi and into a vesicle, and eventually it ends up being displayed on the plasma membrane of the infected cell. So that's how you get these class 1 MHC antigen complexes on the cell surfaces. And the immune system is going to recognize this MHC antigen complex. Now, for the class 2 MHCs I mentioned, they're only found in antigen presenting cells and lymphocytes. And in this case, if this is a macrophage, so macrophage engulfs the pathogen, whatever it may be, and then a lysosome is going to come along and it's going to eat this up. You're going to get these antigenic fragments out of it, which get bound to a class 2 MHC protein, which is going to migrate to the plasma membrane and get displayed. So more or less the same mechanism. The basic idea is just you're going to display the antigen on the plasma membrane surface so that your immune system can detect it. And whether or not you're using a class 1 or class 2 MHC to do it just depends on what cell type are you. So, inactive T cells that have not been activated yet, have receptors that recognize MHC proteins that are bound to antigens. If it's the specific antigen that a T cell has been designed to detect, it will bind. The T cell will bind here. This is called antigen recognition. The proteins involved in this process are called CDs, clusters of differentiation. There are hundreds of them, but the most common are CD4 and CD8. CD8 is found in cytotoxic as well as regulatory T cells. We just call them CD8 T cells. And this responds to MHC class 1. CD4 is found on helper T cells, and it responds to MHC class 2. So CD8, MHC class 1, CD4, MHC class 2. All right, so antigen recognition prepares a T cell for activation. Full activation requires exposure to a specific stimulus. Something called co-stimulation is generally required. And this is where the T cell binds to the stimulating cell at a second site, and this confirms the activation signal. So T cell has bound here, and you get co-stimulation. So some other proteins are going to bind in a second site, and that will confirm the activation. And you can see here this is labeled a class 1 MHC, and that tells us what type of cell the T cell is. It's a CD8 T cell, and it also tells us that the target cell carrying the antigen is probably just an infected cell from the body. It's not an antigen presenting cell. So T cell binds to the stimulating cell at a second site, confirms the activation signal. Following activation, the cell will divide repeatedly, the T cell, and it produces 3 different types of daughter cells. We have previously defined memory cells. So these cells remember the encountered pathogen, so you can mount a quick response in the future. And you also get cytotoxic T cells, which will seek and destroy any cell in the body carrying this antigen. And they can use a variety of methods to do this. They can destroy it with perforins, which we discussed. They can release cytokines that cause the cell to destroy itself through apoptosis, or they can disrupt the cell via the release of lymphotoxins. So it has a few options. But regardless, cytotoxic T cells are your seek and destroy cells. And finally, the third type of daughter cell you get are regulatory T cells. And these just moderate the responses of other T cells and of B cells by by secreting inhibitory cytokines called suppression factors. These regulatory T cells generally don't do anything until after the initial immune response because they take a very, very long time to activate. So I just want you to note that from the appearance of an antigen in the tissue, so like a virus or a bacterium, all the way to widespread destruction of the pathogen takes 2 days to complete. During those 2 days, the damage or the infection is spreading. However, if you get through it and the same antigen is found a second time in the future, memory T cells, immediately recognize it and differentiate into cytotoxic T cells. and that produces an overwhelming and effective response. This is kind of the entire premise behind vaccination. You're going to go ahead and expose the immune cells in your body to an antigen, and your body is going to create those memory cells for that antigen so that if you encounter the pathogen later, you're good. And the trick for a lot of our vaccines is actually trying to figure out what the antigen is, because especially in the case of viruses, mutation is happening all the time. And what the antigen was last year for a virus could be completely different next year. It can even change after it passes from one person to another. You can get a mutation, and all of a sudden the vaccine, which had the antigens you injected into someone, all of a sudden those memory T cells, not as effective because the antigen has changed. So, antigen presenting cells can stimulate CD4 T cells and produce helper T cells, which these promote B cell activation and antibody production. So the T cells must first be exposed to antigens on MHC class II proteins. You're going to have co-stimulation, which then completes their activation. Then the T cell undergoes a series of divisions to differentiate into active helper T cells as well as memory cells. Very important. When a B cell encounters its specific antigen, it prepares for activation, what we call sensitization, and during this time the antigen is brought into the cell via endocytosis, and it appears on the cell surface with MHC class IIs. Which you see here, sensitization. Now, after sensitization, to become fully activated, the B cell has to encounter a helper T cell that was already activated by the same antigen. When this happens, they will bind, and the T cell will secrete cytokines that promote B cell activation. The B cell is then going to divide, yielding memory cells and plasma cells. And it's the plasma cells that secrete antibodies. These cells can secrete 100 million antibodies per hour. Quite a lot of them. This is an antibody. You don't have to memorize the structure. We also call them immunoglobulins. They're just small molecules that bind to specific antigens, and their abundance increases dramatically with later antigen exposure. The rough structure of them is just one pair each of light chains, which are here in blue, and heavy chains, which are in purple. One segment of the antibody is always the same for any antibody you have, and then you have a variable portion of the antibody that can change. And this is just showing you that when an antibody binds to an antigen, it forms an antigen-antibody complex. I don't expect you to memorize what a hapten is, so you don't have to worry about haptens and carrier molecules. You also don't have to memorize these. This just shows all the different ways that antibodies work. Antibodies do a lot of different things, and they have a lot of different ways of dealing with pathogens. They basically just stick to the pathogen, and then they'll choose what method they want to use to get rid of it. And finally, this is just a summary slide of the immune system, how it works. We talked about innate immunity. We talked about specific adaptive immunity. And within that, we talked about class 1 MHC activation and class 2 MHC activation, which cells get activated, and what the end result is.